Today is Altaf Wani. He is a professor emeritus in the Department of Radiology and the Department of Cellular and Molecular Biology at The Ohio State University. In 2004, he was elected a fellow in the American Association for the Advancement of Sciences, and from 2006 up to the time you retired, or perhaps up to the present time, you were director of the Molecular Carcinogenesis Laboratory. So quite a career with a number of publications in absolute top shelf journals. He's a co-author of so many articles that he's internationally known. You can find him on Wikipedia. That's when you know you're famous nowadays, right? And so he's had a full career in expertise in cancer research. So let us please welcome Dr. Wani, who will speak to us with one of the most interesting titles to ever come along in this lecture series, Correcting the Blueprint of Life. Please.
Thank you, Joe. I have had a quite active career, but I am proud of the fact that I'm in my 41st year here at Ohio State University. But I'm in a company where I think 41 probably doesn't mean much. I just met somebody who just completed 50 years. So anyway, I'm delighted to lead off this lecture series and talk about my research endeavors, which happens to be in the area of cellular molecular cancer biology. So along the way, I'll be talking about maybe a little bit of history and mystery of DNA repair. The topic obviously is complex and is very technical. I will try to make it as simple as possible. To get started, I would like to find out if there is anybody who has not heard about DNA. So that's very good. So now DNA, to get you oriented with my talk, this is the structure of DNA, which was identified in late 40s and in early 50s, 1953 structure, deciphering of structure by Watson and Crick got a Nobel Prize. This is a small piece of DNA shown here. It has two strands, and they are going in anti-parallel manner against each other. And the DNA is obviously called deoxyribose nucleic acid, and the reason it is called deoxyribose is because the sugar backbone. So it has a sugar phosphate backbone, and this sugar is a deoxyribose sugar. It lacks one oxygen and is on the second position, as opposed to RNA, which has a hydroxyl group. And so this is the backbone, one strand going in 3', 5', 5', 3' direction. And there are bases, which are these nitrogenous bases, 4 bases: adenine, thymine, guanine, cytosine, which actually form only 4 bases in a sequence forms the basic code, what we call the blueprint of life. These bases are paired in a proper manner against the corresponding complementary base, adenine with thymine and cytosine with guanine always. And these are hydrogen bonded. These are kept together by hydrogen bonds, and the 2 strands stay together, but they are easy to melt apart and come apart as needed by the biological function which may be taking place on it. So this is not to intimidate you, it is just to give you an idea, although the DNA is the keeper of genetic information and it is supposed to be stable, but it lives in an environment where it comes in contact with a whole series of chemical and physical agents, and which range from reactivity to very simple, even molecular oxygen or water can be able to interact with DNA and cause actually quite a bit of havoc. to various chemicals sitting in a couch or sitting in your chair, which are laced with chemicals all the time. And of course, in our environment, ultraviolet radiation, which comes naturally, just we walked through the campus and got a good dose of radiation, or voluntarily just taking puffs of smoke into our lungs and those causing damage to the DNA. And it causes basically the modifications atomic at the various atoms in the DNA, and these can be very, very subtle. And these subtle differences in the structure of the bases and DNA has in anthropomorphic terms over the years referred to as DNA damage. Now, as you can see, this chart here shows different lesions on the DNA, but also in this cell there are all kind of processes and molecular actors which interact with DNA and evoke a response. And that is kind of captured here. So the source of the damage can be from within, like metabolism, the breathing of oxygen, in mitochondria, oxygen is being burned, and the free radicals form the damage to the DNA. Viral infections, radiation, chemical exposure, replication errors, all cause damage to the DNA. And the DNA damage, once it is present in the cell, it is quickly sensed by very specialized molecules called DNA damage sensors, which are also part of the repair response mechanism. And there can be, which can cause cell cycle checkpoint activation, so the cells are not able to divide, transcriptional activation, some new proteins are being formed, and the cells, if the damage is too much, is made to cause suicide, a programmed cell death, which has been referred to as apoptosis, and of course DNA repair, which has been my specialty. Now, DNA repair is important. If you do not have DNA repair, you can have lesions in the leftover lesions in the DNA, and then they can lead to all kind of problems. And one of them is obvious, is cancer. Cancer is result of the unrepaired DNA damage. So you can have damage within the cells, and there is the genes which are part of this process of the cancer development process, the progression process. For example, you can have oncogenes or tumor suppressor genes. Oncogenes can promote cancers, tumor suppressor genes can prevent cancer, and if they are not functional, they can create problem, and the lesion may be anchored and fixed in the cell, which can then, that cell can keep on dividing and become, say, into a state of cancer, shown here with the red group of cells. And normally immune surveillance system in the cell, we all have cancer cells in our system all the time actually. And our immune surveillance system keeps it under check, and actually they are natural killer cells which get rid of it. But those which escape, or you can have cancer which is actually in the clinical setting treated with various therapeutic agents, then these cells disappear. But sometimes from the group of the cells, there may be a second kind of a second type of a cell, secondary cell, which can emerge, and that then becomes very recurrent to treatment and becomes the source of recurring cancers. So that gives you an idea about the significance of DNA damage and significance of repair in the normal healthy state of living organisms. And DNA repair is present in all kinds of living organisms in the plant kingdom and animal kingdom, everywhere. So when the damage happens, I will focus now on, instead of other processes, only on DNA repair. So you can have a base which is not properly paired. Let's say adenine is supposed to be paired with thymine. It is instead of pairing with cytosine, so it's a mispairing. So you can have a mismatch, and that has to be removed. Or you can have a break in one of the strands, and it should be a single strand break, and that is there's a repair mechanism for that single strand DNA break repair. Or you can have actually a more lethal two strands being cut, and you can have a full-fledged break, which can be a more lethal kind of a modification. Or you can have a base, and then base has to be excised and repaired, and there's a mechanism for that called base exchange repair. Or you can have a full nucleotide which will be damaged and needs to be removed. So once these damages are sensed by these molecules, they basically recruit all kind of actors, and the ones who are specialized for specific function, and they come in a very ordered and coordinated manner to the site of the damage and then do their thing. So DNA repair in 2015 got the Nobel Prize. It was the work of many people who actually had contributed to it, but the credit was given to three scientists. Thomas Lindahl from UK, originally from Sweden, and then Paul Modrich from Duke, and Ajit Sankar, a Turkish-American scientist from University of North Carolina. I have interacted with all three of them. I had a privilege of interacting with all three of them, but my research has overlapped that of Thomas Lindahl and Ajit Sankar. Thomas Lindahl actually in 70s actually was able to provide guidance for my doctoral research. And then Ajit's research and my research has actually paralleled over the years because he and I work on the same nucleotide exchange repair process. So Thomas Lindahl was recognized for base exchange repair, Ajit Sankar for nucleotide exchange repair, and Paul Modrich for mismatch repair. So three scientists, and I'll be talking about the nucleotide exchange repair because that is that has been my major focus of research. So one of the things which actually one takes away from this Nobel blurb is that the life exists, so DNA must be repairable. Obviously, you can see that there are almost normal metabolism in our body. The estimates from Lindahl actually, there are almost 10,000 bases lost per cell per day. And you can imagine how many cells are we having and how much bases are lost every day. So that's a normal metabolism. There's so much damage. And then from exogenous sources, there is a lot of damage. Now, if that damage exists, obviously DNA cannot be considered to be genetic mutated. It will not function. It cannot be stable. We will not live. So it must be repairable. And over the time, it has been proven that we do have a very efficient and well-coordinated repair mechanisms to take care of the insults within our DNA. And second thing which I would like you to take away with is that as soon as you have DNA damage, it is not one or two proteins. Actually, there are swarms of proteins which congregate at the site of the lesion in a very prompt manner and actually are having various sophisticated functions to be able to accomplish the productive task of making repair happen. So then obviously I will not be able to talk about the research, but I will give you some kind of idea about the work which we have done. These are sequencing gels, which actually is another area which got Nobel Prize in 1980. Fred Sanger from UK and Wally Gilbert from Harvard got a Nobel Prize for identifying these sequences where you can be able to read sequences of the bases because in these gels there is a size difference of one nucleotide at a time. You can be able to read, for example, these are C's, there will be T's, and so on and so forth. And we have been used it in 90s as an assay to monitor repair at single nucleotide resolution. So, and this is another thing which we have been using, which is based on polymerase chain reaction, which actually got Nobel Prize in 1994. Kary Mullis, actually from Stanford, he was recognized for identifying the PCR reaction. Unfortunately, as a young scientist, he died last month. So anyway, we have been able to use this polymerase chain reaction to be able to amplify these small areas of the DNA, and this is a small segment of the DNA which we are trying to monitor the repair. And here is the sequence. For example, this is the control shown here, and we have been able to, wherever there's a lesion, we have a mechanism to be able to cause a cut with an enzyme to be able to show if there's a cut and there's a damage, it shows up as a band shown here. These are duplicate lanes, as you can see, as a function of time after introduction of damage. This is control. As you introduce the damage, at 0 time, you can see a lot of damage as an indication of a damage. Presence of this band indicates the damage. But as a function of time, you can see this band disappears, and that's a repair taking place. So within 24 to 48 hours, repair has taken place at this particular site, at this codon. And you can see it all along the codons. And one thing you can also notice is that at some sites, although damage is occurring, but repair is not as efficient. Repair is less efficient. So you can see at this site, you can see the repair here is very, very fast. Repair here is very, very slow. And interestingly, it turned out this is actually a segment of the tumor suppressor protein p53. And if you look in the clinic, 50% of all tumors found anywhere in the world have mutations of this particular gene, p53 gene. And when they look at these mutations, they found there are hotspot codons where these mutations take place. And for example, this turns out to be one of the hotspots. And the reason it's a hotspot of mutations is because at this site, the repair does not take place. So if repair does not take place, and that is the site where the mutations will accumulate, and those are the ones which actually show up as tumors in 50% of almost all tumors in the populations. So this is the example of looking at repair at a single nucleotide resolution. Next, please. So, and there are different rates of repair. And this is under the conditions where actually tumor suppressor protein in the cell is dysfunctional. This is a wild type p53 in the cell. The repair takes place normally. But if this p53 is dysfunctional at the same exact site where there is repair is taking place, no repair is taking place. You can see the bands are still intact and they are all over the place. So it gives you an indication in a cell there can be the absence of a repair protein can lead to defective repair, or the absence of a controlling mechanism, a regulatory protein like p53 can lead to the dysfunctional repair and obviously the repercussions of lack of repair. And tumor suppressor p53 actually is known to be the guardian of the genome in the cells. It actually directs many different processes in the cells, and that's why we kind of focus on that. So this gives you an idea about the repair, how we have been looking at it, and we have been able to look at it in 90s. In fact, 80s and 90s, we have looked at DNA repair under all kind of scenarios and all kind of cell types, normal cells, mutant cells, tumor cells, and engineered cells, and so to be able to understand the mechanism of actual repair process taking place within the cells. And still we are talking about only the nucleotide action repair. Then there are other people working on other types of repair. Now, I'll quickly switch gears now. Oh yeah, voila. So we looked at, when we were just looking at the repair in 80s and 90s, we'll just look at the cells and tissues of animals and so on and so forth, and grind them up and isolate the DNA and analyze the DNA, just one of the examples I gave you a little while ago. But normally, we wanted to see what is happening inside the cell in the normal environment of the cell. And there was really not many techniques actually available. There was one technique which came in from University of Wisconsin by one gentleman to look at in the native environment of the cell. And I tried to adopt that technique, and it was very cumbersome, and other people have not also been able to follow that. But the reason you have to look at within the native environment of the cells, because DNA is about a couple of meters long, but it is actually put into a small tiny space of about 50 microns, and it's all wound up and it just sits in the cell. So how actually the repair machinery is able to access these different damaged areas within the genome obviously was a mystery, and we wanted to figure it out. Now, in cells, you have this very specialized kind of a way of packaging this material inside the nucleus. You can have these nucleosomes, and then you have these bead structures, and then there is other superstructures, and ultimately in the form of chromosomes, it is packaged into this tight space, and it is very well organized. And when you have the repair machinery going to go and find, say, damage, I think it is responding. So then you have to the repair machinery has to overcome the constraints of this packaging. So we were fortunate in around 2000, we got this technique. There was this group of Japanese scientists and the Dutch scientists from Erasmus Institute, which is considered actually a mecca of DNA repair research. And they came up with this technique. And I have been thinking about this problem for some time, and we kind of jumped on this, and my lab was actually primed for this kind of research, and in 2000 we started it, and we are almost for the last 20 years exploiting this kind of approach quite a bit. And the technique is based on very simplified kind of a mechanism where you just are able to take the monolayer of cells and you overlay them with these filters. And these filters have pores in them, micropores. This is about 3 micron pore and 5 micron pore. And obviously when there's a 3 micron pore, there will be more holes. If you just overlay on cells, you'll have more areas through which you can be able to shine this ultraviolet radiation to cause damage. Then you have a larger pore filters, and then you will have a smaller area. So I had actually in 80s as a postdoc here at Ohio State developed these antibodies which were able to specifically recognize DNA damage. And I had done quite a bit of work using all kind of readouts but numbers and so on and so forth. And we were able to see repair, and we have obviously published on that. But this was the first time actually I felt very satisfied to see that I can visually with my own eyes see damage. So if you have, and these antibodies are DNA damage specific, not protein specific, DNA specific, and they can be able to bind to the DNA damage. And these red areas are shown here. This area has been flooded with antibodies, and then after processing and just tagging them with the fluorophore, in this case a red fluorescent tag, you can see this is the nucleus here. Oops. This is the nucleus here shown in blue, and this DNA. The only difference is that this area has seen the light through that hole, and that means it has the damage. Antibodies are only able to recognize that. Obviously, I don't need any convincing to tell you that these are DNA damage specific antibodies which are able to see it. So then, so we have been using this technique and using the DNA chromatin. And also there are antibodies to proteins. The proteins, all kind of proteins, which are commercially available now. We have developed some on our own. And these proteins are repair enzymes, which are able to go to the site of the damage. And if you have no damage in the cell and you try to label the cells with these antibodies and then look at the binding, and this green is showing that this protein, repair protein, is present in the cell, in the nucleus. But it is all over the cell, everywhere. But if you just now introduce the damage, just like this damage, and then use two different colors and then to see interaction between the damage and the repair protein, you can be able to see that immediately, actually this protein, which is all over in the nucleus, congregates at the site of the damage. The red underneath is the damage, and the green is the repair proteins congregating like a comet to the site of the lesion. We normally do in our lab 10-15 minutes after the irradiation. But some people have techniques which are actually using live cells. They show that these proteins can go in about 20 seconds, go to the site of the damage, and then are able to do their work to repair the lesion. And it's not one protein. There are dozens of proteins, even more actually, scores of proteins, which congregate at the site of the lesion, and they have different function. And that's what we have been able to tease out over the years. And this is example, I use it to show to these undergraduate students to make a point, and I thought it will be also useful here. I was in a meeting in China, Wuhan, and they took us for this acrobatic show. So I started shooting some pictures, and then I was looking at these pictures, and I just thought, okay, maybe this is something I can use it to relate what I do. So let's say this is the auditorium. This dark area here, this is the cytoplasm in the cell, and this big stage shown here. This big stage here, shown in blue, is actually the nucleus of the cell. And then you have this here smaller stage, just like that damaged spot, which actually we introduced through these micropores. And then all these acrobatic molecular actors come to the site of the lesion, and they do their thing. And there is the mitochondria, there is the oxidative phosphorylation going on, there are other habits. And this one, I said, okay, this was the guy there. I said, okay, this is P53, tumor suppressor protein. It is basically directing these molecular actors to the site of the lesion. And you can see that these young acrobats come, do their things, come in a sequence, and then just are able to do the whatever acrobatic feat they are able to do, and it is perfectly working. And it's like a normal cell. In normal cell, all these factors will come in a normal coordinated sequence and be able to do the thing, and the cell is very happy. But what if there is something missing, if there's a repair deficiency in the cell? So one of the girls is not around or is sick. Obviously, you will have the repair will not take place. This acrobatic feat will not take place. Now, it depends upon which girl is sick. If the last girl is not around, you can have all these girls come up to the site. They will be there, but they will not be able to perform the task. You will not be able to see the acrobatic feat. But you will have the rest of the pyramid will be formed. But what if this girl, which is supposed to be the first one to come to the site, if she doesn't come in, nothing will be able to congregate at the site of the lesion. So you can be able to have a defective repair. And this is the approach we have used over time. And we have this technique called RNA interference, where you can be able to knock out any factor, any protein in the cell in a very specific manner. And we have been able to do that and then ask questions which comes first, which comes second. If first comes, then it doesn't, what happens to the function and what happens to cells. So all kind of questions we have asked over the years to be able to form a mechanistic answer to our queries. So with that, I think I will basically conclude. I will not take much of time. And here is the thing. Over the years, we have been able to, on the basis of this kind of research, we have been able to show that it's a very complex phenomenon. So if you have damage, there is the strand-specific repair. There's a transcribed strand and non-transcribed strand. Different players are able to recognize the damage at the site of the DNA. And then different players, for example, is a DNA damage binding protein, which is acting as a sensor of the damage. In this case, RNA polymerase is sensing the damage. And then they are able to congregate. There are many things which are common. Other factors are common, not only within the repair process and other processes within the cells. So after these sensors are able to come, they latch onto the DNA, latch on the damage site. But if they latch on, then they have to come out to make room for the other factors to come in, so that, and there are processes like this ubiquitination process, which is able to cause the modification of the initial sensors and make them vacate, so they hand over the lesion to the next series of proteins, and these are sometimes degraded by recruitment of the proteins, protein messengers like proteasomes and so on and so forth. They degrade the proteins. So ultimately, the idea is you can have the ubiquitination, you can have simulation, you can have phosphorylation, you can have ribosylation, and so on and so on. These are other layers of DNA regulatory mechanisms. The ultimate goal is to be able to form this pre-incision complex in such a way that this lesion piece, the damaged piece, is picked up in an effective manner to achieve productive repair. If any of these functions is defective, which can be naturally or you can be able to induce it, repair will not take place, and you can be able to measure that. So that's what we have been doing for last 40 plus years at Ohio State University, and it has been a very satisfying experience, and you can see that it has been a back-breaking job, but it has been a labor of love. I wish I had another 40 years to carry out and just dig in and just peek into these kind of processes some more. Obviously, now I am an emeritus. I'll have to look forward to these special kind of discoveries from my younger generation of scientists. And I think with that, I will kind of conclude, and I hope I have not taken too much of your time, and I'll be happy to answer any questions.
Time for questions and answers. I'm going to have the mic, and I'll walk around
Okay.
so everyone can
Yeah.
hear.
Absolutely.
That is, I don't think I'll ever look at Cirque du Soleil the same way again. And so that is the incredible part of this. So let's have questions for Dr. Wani, please. And I'll pass the microphone on.
Yes.
If the repair mechanism is defective, has your research shed any light on how to fix repair mechanisms?
No. There is no way of, there's no treatment where you can be able to, if somebody is having a defect, you can use preventive measures. One of the things, for example, UV, this disease which is called xeroderma pigmentosum, actually these patients are susceptible to sunlight. They are sensitive to sunlight because of the UV. They don't have actually ability to repair their UV damage in their cells. And they actually have sensitivity, and they develop cancers early on. So you probably know that they are not asked to be outside in the sun. They only move around under the hood sometimes in the night time only. That's a preventive measure you can provide. There has been actually attempt, but it has not kind of worked too well. There was actually some people from Long Island, a company from one of my early peers who actually tried to take these DNA repair proteins and put them in creams so that they can be able to put it on the skin so that you will enhance the DNA repair. So, and there was actually a cream which was sold by Astrolade some time ago. For $35 bottle, it was said at Lazarus called Night Repair. And actually, our lab was actually involved in looking at the research, and we used to these students and shine UV light on their one butt cheek, and other one will not take samples out of that and put cream on one, not on the other, and then see if there was any helpful thing happening by the application of the cream. We did not find any different. There was some kind of a hyaluronic, it's a very ordinary kind of things in there. But Astrolade, they were selling it. People probably were buying it for $35, a small bottle, selling at Lazarus. But actually, there is no repair mechanism. There are people who will kind of say certain kind of natural compounds may be able to help the repair process or prevent actually the formation of damage. That probably is more so effective than actually finding the repair. So yeah, no, there is no mechanism to help people with defective repair.
Thank you, Dr. Wani, for such a seminal presentation on what goes on with DNA repair that I wasn't, completely aware of. But I have just a couple basic questions. It appears to me that this is a highly, very, very, very complex molecular machine. I mean, could you categorize this as a extremely complex molecular machine, biological machine?
Yeah, so it's a very powerful question. In fact, so nature is not really extravagant. I mean, nature will like to do things in a very simplified manner. And we actually were asking, we identified this process, and I was kind of puzzled by it, and we found this process of ubiquitination. As soon as the sensor comes to the site of the lesion, and then actually it gets ubiquitinated actually, and it puts the morph, and it shrunks the body because these things move around, and then when you have ubiquitination, it actually binds, it latches on the damage, and then other pieces come on. Then I found out deubiquitination, which was also very important. There are deubiquitination, and we have been working on it actually lately quite a bit on that deubiquitination. So basically there is ubiquitination and then deubiquitination. So if you want deubiquitinate, why then you want ubiquitinate? I mean, it does not make sense. If you want to take off, then why do you put it on in the first place? So then it actually was kind of a vexing question for us, and we were able to conclude what happens is if you have ubiquitination, it wants to keep it for a certain period of time at the site, and then you have to deubiquitinate so that it actually is able to vacate from the site of the lesion. And it has to be there for a certain period of time. If it comes off prematurely, then you don't get productive repair. So there is obviously a lot of complexity. Some of it we understand, some of it is not. And it has been obviously bothersome. These questions keep on coming up, and then the thing has become so complex. It's an alphabet soup, my research in alphabet soup. When I try to sell it to the reviewers, and normally you don't get reviewers who are probably as knowledgeable in your area of research, and it actually overwhelms them. And then they slap me around by throwing the approach problem and this problem and that problem. I've been fortunate to be funded for 30 plus years continuously, and my funding continues until 2021. And I have actually, my proposals now are a lot better. I am very happy to see my own proposals, but they don't like them. Because I realize, what's wrong? This looks a wonderful thing. So it is a complex process. And like I said, actually, there's probably even more complexity which we have not unearthed yet. And it's going to be fascinating. But that is how nature is. Yeah.
Next.
Yes.
Well, thank you. I have a very simple question. In a very complex process you have shown, which we partly understand, can you take some of the known drugs that we are aware of, and can you show the specific site where they can interact?
In the realm of damage, I mean, with the DNA?
Damage or
Yeah.
I'll give you an extreme example of what happens in sickle cell anemia. And cells get all, horrible shape. Well, okay, that's one. We know what happens morphologically. But now with the biochemistry, repair, and so on, because some of these drugs provide very important tool to understand the process. and I think one should take advantage of that.
No, actually, no, you're right. Actually, that is actually a very active area of research. Quite a bit. People are trying all kind of agents and have been able to look at the modulation of repair. Actually, most of the time to help the removal of the damage at a fast rate and to prevent the you can look at the death of the cancer cells versus no death of the normal cells. So differential actually killing of the. So these agents, there are a host of them which have been tested with the aim of targeting the tumor cells specifically. So, and there is obviously interaction, and they do interact with the repair systems and then actually affect the downstream process like checkpoint activation and so on and so forth to help the normal cells but not the cancer cells. And there are so many. One of my areas of research is looking at the natural compounds, and one of the compound which I have actually worked on quite a bit and published on that, some of the graduate students have worked on that, is this compound, this colonji, which is a black seed which has an active component called thymoquinone. And thymoquinone we have been able to use in the lab to show that actually it is able to target the tumor cells better. But if it affects the repair or it affects something else, we don't know. We feel that it has certain other properties where it actually affects the oxidative damage and this regulation of oxidative damage, and therefore prevents the normal cells from dying and tumor cells from and kills them and primes them. And there are similarly many. We just also another thing, because you are from pharmacology, Dr. Patel, right? So one of the things which we have actually used is naringin, which is actually from the citrus peel. I mean, you see that kind of thing if you just squeeze the skin of the citrus, and you can see that, or that's a naringin. And we have actually published a very effective paper in cancer research, in which normally the cells which are not dying to cisplatin, there are these ovarian cancer cells, they become resistant to cisplatin. Normally, if you take the ovarian cancer cells, cisplatin kills them. But once they become resistant, they don't die. Cisplatin doesn't kill them. So we were able to take this naringin and combine that with cisplatin, and we show that even the resistant cells are able to die now. And there's a mechanism we have shown that actually it primes the cancer cells, develop a kind of a mechanism to survive, and this naringin pushes them so that they are not able to resist to the treatment of cisplatin. So those type of interactions with all kind of compounds have been tested. It's a very, very active area of research, and I would say thousands of researchers are working on that.
I'll take the mic all the way back there. My question is a little different. When you go to an oncologist, the question I would ask now is, what is the training of the oncologist in regard to this? How should oncologists be trained, given this is a very deep, basic area of science? And how long would it take to really train an oncologist to really know about things like this? I see the significance of what you're trying to do. So I think there's a question there in the translation between the basic sciences and what might go into medicine and what the treatment might be. So that's the kind of question.
Yeah, so that is actually also happening. Obviously, the oncologists have their hands full. They have all kind of things to train. These days, what is actually big focus on is immunotherapeutics and so on and so forth. But in the case of this DNA repair, we have been training our radiation oncologists, those who are practicing radiation oncology, where they give the ionizing radiation to treat tumors. And we are able to, in the classroom setting and also in the research setting, able to train them to understand actually when you use ionizing radiation, actually how actually it is able to kill cells. And if you are able to have a defensive mechanism like a repair mechanism, why your ionizing radiation treatment can fail. And if it fails, so how can you make it successful by using the agents like these chemosensitizers or radiation sensitizers to be able to sensitize them to the radiation, and because you are able to compromise the repair mechanism, so when you just treat them, and that is actually practiced quite a bit. So in radiation oncology and in I would say medical oncology also, people are getting well versed. I mean, many of these scientists are, as you can see that Ajit Sankar, he's an MD, as a matter of fact, Thomas Lindahl, he's an MD. These are, and I have actually, by my education, I'm a PhD scientist. But I have a really lot of respect for the MDs who are doing research, because they are able to imbibe all these concepts and actually take it to another level. And they do that very successfully. And a lot of these scientists who are MD scientists devoted to research, basic research or applied research, they do a wonderful job and do practice the kind of thing you are propagating, you are promulgating.
Thank you again for the wonderful talk. My question is maybe related to the previous one, but maybe the flip side of it. As you pointed out, cancer, let's say, starts by some sort of an attack on the cell and smoking, let's say, in the early days. And then the body comes to try to repair things. So we're naturally led to ask, what makes that repair kit as strong and robust as possible? If you watch enough late night television or enough WOSU, you see shows that purport to tell us optimal diets, which presumably are working on robustness of the repair kit. What's your evaluation of recommendations made to the general peon audience, of which we're part, the non-specialists, about what we should be doing and what we shouldn't be doing?
As you can see, that the repair is very complex. We have understood a lot of it, and a lot of it remains it is undiscovered. So why will you tinker with the repair kit, or if you want to go basically do that? So only thing we have been, and the scientists have been able to help the individuals, is by telling them we cannot, because everybody has a specific repair capacity. In this room, there are people who have mechanisms, same mechanisms to repair, but the levels actually differ. For example, in the case of women get breast cancer, for example, some get, some do not get. People who smoke, some get cancer, some do not get. And there's a threshold, some threshold level of oxidative damage which is happening. And then certain amount of that is able to, bodies of the women who are able to repair that or able to cope with it. If it goes beyond that threshold, it goes above that repair capacity, the damage starts accumulating. So you cannot, and those are the ones actually, the women who will be able to get breast cancer because of the lack of repairability. And it doesn't have to be, it has to be, they have a repair system. Some do not have totally defective repair system. We know BRCA1, BRCA2, now there are these mastectoidomies and so on and so forth, prophylactically and so on and so forth. But individuals have, similarly for the skin cancer, similarly for other cancers, every body is equipped with the repair kit, the capacity varies. And depending upon the degree of capacity, some are prone to be able to manifest that damage in the form of mutations and the tumors, some are able to avoid it. So from the repair standpoint, people have not been given any risk. From the damage standpoint, there has been, so you can be able to take the agents, your diet, for example, your other behavior. Smoking obviously is obvious one. You don't smoke, you don't get cancer. You stay outside of the people like to go and they just kind of like to sit in the sun. You just stay away from the sun. Say late afternoon sun, which is very bright, has a much higher UV component and therefore has people who are doing tanning can obviously get cancer. So this is a very simple suggestion. Then the food, as a matter of fact, there's a big thing, probably the area of, so Dr. Patel was referring, huge area of these phytochemicals. You can take wine, which has resveratrol. You can take isothiocyanates. You can take the red berries. You can take all kind of things. You can take pomegranate juice and all that. And what they are actually are green tea polyphenols. Actually, green teas. Tomorrow noon we have a lecture from one of the experts coming from Case Western Reserve University in the urological cancers. Green tea. They have worked on green tea. So they have polyphenols. What they do is, as a matter of fact, they prevent the oxidative damage. These are scavengers kind of thing. So if you have loaded your cells with these agents, you can be able to prevent the damage. And one of the damage, I am a believer of the fact, although it is not my primary area of work. I work on nucleotide excision repair. The oxidative damage which is happening in our body, oxidative damage is a source of not only cancer, aging, and other degenerative diseases and so on and so forth, because we are subject to a huge burden of oxidative damage. If we can keep that low, one way to do that is just taking these compounds. You can take a ton of garlic, and garlic can prevent from having oxidative, but you cannot take too much garlic, you will lose your friends. But but you can take pomegranate juice. How much pomegranate juice? One of the biggest problems with this area of research is we can demonstrate they work beautifully in the laboratory setting. I can show it in the tissue culture cells. It actually makes the cells look healthy. Cells are able to survive longer. There's not many transformations to tumor cells. But in the real setting, it does not seem to work because the dose we use in the lab is relatively higher, much higher dose, and we can be able to show the effect. But the amount of dose we require, we need to take a lot of these berries. We have to eat berries whole day long or other things whole day long, and we cannot get it. So there is some, but main thing is actually the oxidative damage can be prevented. So similarly, other damages can be prevented. But repair, you cannot be able to. You can probably take, there are people are kind of promoting this and that increase your repair capacity, but there is no substantial evidence to show that I can be able to help with your repair. Repair is what you have, you have.
We have time for one more question, and it's right here.
Is it clear why a response to a perturbation is apoptosis versus some other pathway?
If so, apoptosis versus
Some other pathway, like necrosis?
Yeah, so it depends upon the degree of insult within the cells. Now, necrosis you have to have, it's a pathological kind of a thing, and it is happening in response to some kind of a major insult to the tissue, and you can have necrosis. But apoptosis is a much more controlled phenomenon, and apoptosis is kind of a mechanism where cells fall off. Apoptosis in Greek means falling off. Cells just fall off. We have, when we just grow up, we have developmentally we have webs like this, and then we just have fingers, and the cells in between just go into apoptosis. It's a programmed cell death. Actually, then we get our fingers initially, but these cells are prompted to die off in a programmed manner, and it happens. And so apoptosis is also as part of the cancer process. Cells die through a mechanism of apoptosis, and they do that. It's actually a defense mechanism, a repair mechanism. Now, if you have a certain amount of damage in this cell, that cell can live. Repair mechanism will come and repair it, and then the cell will live. It will be fine, because repair has been completely damage has been completely repaired. But if the damage is too much and the cells are not able to repair it, then the cells automatically invoke this programmed cell death, which we call apoptosis. So it's a defense mechanism. That cell, which can be a problem, is eliminated from the system. So the cell dies. It does not become a source of problem like developing into a cancer down the road. So that is also happening on a regular basis in our body. So we may go into the sun, we may get damaged cells and repair will take place, but also apoptosis all this at the same time makes those cells disappear. One of them is obviously we slough off quite a bit after a big sun tan, but the necrosis obviously is much more severe kind of a response to severe injuries.
Let's give a